Thank you so much for having me today. I'm really excited to be here. I will be talking about my kind of first publication I'm hoping to get in my PhD, and it's about whale earwax. But before I get into that study, I am going to tell you a little bit about how I got to where I am today, because I think it's interesting and hopefully helpful knowing that you don't have to take a straight line to get to where you want to be. So eventually I will be discussing how I got over 100 years of data, specifically pregnancy data from these animals in order to look at their population dynamics. But first, I want to get into something else. So first off, my name is Dani Crain, third-year PhD candidate, which basically means that I've passed kind of my main exams in my PhD program, so I'm not a PhD student anymore, I'm a candidate. So they're gonna have a hard time kicking me out now. That's basically what that means. And I moved into Texas in August of 2015 from Northern California to start my PhD at Baylor University. And I study whale endocrinology, and endocrinology is the study of hormones. But I'm also a powerlifter. So I wanted to bring this up because we often think of scientists as kind of these stuffy people, don't know how to have fun, they only do one thing and it's nerd out about science. And while I'm very happy to nerd out about science, I also do a lot of other things. I powerlift, I knit, I have a dog I'm obsessed with. So scientists are a lot more varied than sometimes we give them credit for. But I'm also here representing Present Your PhD, which is a program that I helped found at Baylor University. And we're really interested in actually connecting the community at Baylor with the community in our surrounding community, which includes all of you. And we started with the four of us, and now we've grown into an organization with about 15 people, all PhD students that are really eager to share their research. So last semester we came a couple times, and we'll be here myself, and then in a couple weeks you'll have another member talking about his research in environmental science. You can find us on Facebook if you're interested. So how did I get here? Well, first off, like I mentioned earlier, I'm from Northern California, so graduated high school and I went to started my bachelor's at UC Santa Cruz, which is a beautiful, beautiful place, kind of the main reason I chose it. And I graduated in 2009, which was a horrible time to graduate because we had that huge recession. Right, 2008 hit, and then in 2009 there were no jobs. And unfortunately, I had planned on having a job between my bachelor's and eventually going to graduate school. As you can imagine, that didn't happen, even though I applied to quite a few places. So I ended up doing an internship, which means I ended up actually paying them a little bit for me to work for them, which is silly, but sometimes that's how it happens. And I ended up applying to graduate school at Duke University. So I ended up doing a master's there in environmental management. And it's a little bit of a different master's than kind of more traditional graduate school, because a master's of environmental management is at a professional school. So you have 2 years, you're taking classes, and there's a thesis component, but it's not the main focus. And it was very interesting because during that master's I learned I'm not really into management and policy and dealing with the government all the time because it's very bureaucratic. And there's a lot of laws to learn. So that actually helped me, even though I enjoyed my time there and it's definitely helped me. In particular, it didn't help me necessarily know what I want to do, it helped me know what I definitely don't want to do, which is also really important. So that was 2010 to 2012. And then after 2012, I was looking for PhD programs because I was really interested in moving on and really getting into some nitty-gritty science and a more traditional graduate school where I'd be studying for however many years and working towards a PhD degree. But it took me 3 years of trying. Again, this is a 2012, 2013 now. Recession isn't quite done with us yet, and there's not a lot of funding for graduate programs out there at that time. Especially in the area that I had been focused about at that time was more behavioral research. Those are really hard to come by because so many people want to do that kind of work, especially when it comes to marine mammals. We all just want to be out there looking at them in the wild. And so it makes getting a PhD, focusing and looking at that kind of stuff, a bit harder. But I kept trying, even though every time after I applied and I didn't get in, I was like, you know what, this is the last time. I kept trying. But I ended up having a lot of weird jobs in between. So in 2013, I was actually a sales representative for a publication company. They actually publish books on animal, specifically pet behavior, like how to be a better cat owner, dog owner, how to train them with positive reinforcement. That helped me learn a lot about how to interact with my own dog. And it helped me also learn that I'm not going to be happy doing that. So next, I ended up working at the California Department of Fish and Wildlife, actually helping them put together a salmon monitoring program. California salmon not doing great, and I had to wrangle many scientists to try and get them to submit and update their chapters to this monitoring project. And that helped me learn that I definitely don't want to do that either. Trying to get people to do work when they have 15 other projects going on and I didn't have a lot of authority coming into that job was extremely difficult. So after a year, I said farewell, and I ended up becoming an adjunct lecturer at a community college and then also at a four-year university. And what this means is I wasn't an official, I wasn't like a faculty member, but I would come into the university, I would have my class, I would lecture, and then I would leave. And so I didn't have my own office or anything. Really hard being adjunct lecturer. You end up driving a lot to get in between multiple universities and colleges that you may be working with. But it helped me a lot really learn how to teach. Because originally when I was thinking about teaching, I thought, well, I'll just teach how I was taught, which was 100% lecture and sometimes fairly stuffy, to be honest. So that helped me a lot. And actually getting into Baylor, that teaching experience helped impress my advisor, and he knew that when it came to TAing at Baylor, which I do now, that I would be okay. So even though I kind of had steps over here and sometimes a little bit backwards and sometimes a little bit forward, I still ended up where I want to be. And so I had hoped to be in the middle of my PhD, actually be done with my PhD by now originally, but I'm 30 and I still have a couple years. I think that's actually still quite good. And I know if I hadn't had all these experiences, I wouldn't be approaching my PhD in the same way at all. So this helped me grow up, and now I'm here, really excited to tell you guys about my PhD. I did want to mention one last thing here. What are my future job prospects? What do I think I'm going to be able to do after my PhD? As you may know, since many of you are juniors and seniors, you're probably asked, what's your next step? What are you going to do? Right? And that's the most terrifying question. But I'm thinking about this all the time because I know I need to be thinking about it. So I know for me, kind of the easiest thing I could probably do after this is become a lecturer again. And hopefully actually have an office at the university that I taught at. But while I really enjoy teaching, it's not my only love. And so that's why I have faculty up here. So a faculty member, they would teach part-time, but then they would also research part-time. So this is like what my advisor does. He's researching kind of through his grad students, but he's also teaching on the side as well. And finally, I could work at a government agency. And so this appeals to me for a number of reasons. First, the money is going to be a little bit more consistent than potentially faculty could. Because as a faculty member, if you're not bringing in enough money all the time, your job might be in jeopardy. So that's sometimes an issue. But working for a government agency, not as much an issue, although recent government shutdowns, that could be a problem. But the National Oceanographic and Atmospheric Administration, NOAA for short, is often a place that marine mammal scientists like myself will look for jobs when we want to do research but don't want to really do any teaching at all. So that is also an option. And specifically, they have a cetacean health and life history program I'm looking into. And so I'm just always thinking about these things, and every few months I'm like, well, maybe I'll do that instead. And then a few months go by and I'm like, well, no, maybe I'll do what I thought I was going to do in the first place. So I'm always thinking about it, always trying to figure out what's the next step. All right, so in order to talk to you about my project, I think I need to give you a little bit of an intro to endocrinology. And some of this may be, I'm going to say basic, but I just didn't know how much endocrinology you had. I know in high school I had absolutely none. So I'm kind of taking it from that track there. So here are some of the endocrine organs that we have in our bodies, all right? So these are organs that are producing hormones, which are really just these chemical messengers that help communicate signals to the rest of our body. So kind of one of the main ones here is the hypophysis, which is our pituitary gland. And the pituitary gland gets input from the hypothalamus, which is another gland of the endocrine system. We also have the thyroid gland with these little parathyroid glands embedded within that, the thymus gland. The pancreas is a really big one. I'll talk about that a little bit later. We also have these adrenal glands on top of each of our kidneys. They're producing cortisol, or the stress hormone. And there's two main types of glands in our bodies. We have exocrine glands and we have endocrine glands. So exo means outside. So these are glands that are going to be producing something that's going to leave our body. So this would be something like salivary glands or sweat glands. Those are both exocrine glands. So this is an example here of one of our exocrine glands. And then the endocrine glands, these are going to be producing hormones that are just going to travel within our body. And so this is an example of some endocrine glands here. The hormones are going to be released. They're going to get into your bloodstream, and they're going to travel throughout your body and then interact with some of your cells elsewhere. So like I mentioned already, these hormones are chemical messengers. All right, so their job is to travel through the bloodstream and affect certain target cells elsewhere in your body. So if your body is stressed, so if you're stressed out, right, you have an exam, you're stressed out, your adrenal glands are going to produce cortisol in response to that stress, and that cortisol, that hormone, is going to travel throughout your bloodstream, and it's going to actually target certain cells to help you deal with that stress. All right? So this is just an example. You literally have chemicals moving through your bloodstream, right? So your blood is not just made up of your red blood cells, your white blood cells, and all that stuff, but there's also hormones moving through your bloodstream pretty much all the time. So when I mention target cells, I literally mean there's actual cells that have receptor proteins, right? There's tons of receptor proteins in the wall of a cell, right, in the cell membrane. But in this case, each target cell has a receptor protein that is very specific to a particular hormone. So not every hormone can target every cell. All right, so in this case, hormone 1, we can refer to as the ligand of target cell A, because we can see that hormone 1 fits in well to this receptor protein. But hormone 2 doesn't. All right, so simple as that. Now, the reason we have these hormones that are moving through our bodies, their job is to help us actually maintain homeostasis. All right, so who has air conditioning in their house? Right, most of us, it's Texas, right? So if you have air conditioning in your house, it's also working to maintain a certain temperature in your house, right? So if you set your thermostat, let's say at 78 degrees Fahrenheit during the summer, then your air conditioning is working to maintain that homeostasis of that temperature the whole time, right? So if it gets too cold in your house, your air conditioning turns off. If it gets too hot, your air conditioning turns on. This is what homeostasis is. And homeostasis, other examples, so temperature of your body works in the same way. Acidity of your blood. Does anybody know what the acidity of your blood is supposed to be? Yeah. Yeah, 7.2. So if the pH of our blood moves much outside of that, it's actually extremely dangerous for humans. So that's another way that homeostasis acts in our body. We can even think of disease as a way that our bodies maintain homeostasis. You have too much of a virus, too much of a bacteria, your body's going to work to reduce the number of that bacteria, sometimes by fever, sometimes by other reactions. And homeostasis really involves some pretty complex feedback mechanisms. So I mentioned I was going to bring up the pancreas, all right? So the pancreas is the super cool organ. It's beneath your stomach. And it has these endocrine cells that are called pancreatic islets. And of these pancreatic islets, you have two types. You have alpha cells and beta cells. And alpha cells are producing glucagon, and beta cells are producing insulin. So I'm sure we are very familiar with insulin. What is insulin often used for? Yeah, it's used to control diabetes. Type 1, which you're born with, and type 2, basically you become resistant to insulin and you need a kick of it for it to work. So how this works is insulin is going to be secreted when your glucose levels or the sugar levels in your blood are high. Insulin's job is to reduce those sugar levels, all right? And the way it does this is There's numerous ways. I know this is too small. I didn't want you guys to get too focused on the details, but there's many ways that insulin is going to control glucose concentration in your blood. The main way is it's trying to get certain cells to take glucose into those cells so that it's out of your body. All right, and that ends up getting us back to homeostasis, where we have a concentration of glucose that is kind of safe and normal for us. Now, glucagon, which you may not have heard of, is secreted when blood sugar levels are really low. So this might be around 1 or 2 in the afternoon, like you ate, but now it's been a few hours and you're starting to feel really sleepy. All right, that's when glucagon is going to kick in, when your blood sugar levels are really low. And it's actually targeting liver cells and skeletal muscle fibers. Both of those organs or both of those cell types actually will, they're storing glycogen, which is basically just glucose molecules bonded together. So it stores those things. So when glucagon actually targets those cells, it says, hey, we actually need this glucose, please release it. And then those glycogen bonds get separated and then distributed throughout the body, and then you wake up a little bit. All right, so that's again coming back to homeostasis. Now, how do you think we study hormones in humans right now? You can just go ahead and shout it out. Say it a little louder. Activity in the brain, yes. So we have the pituitary and the hypothalamus in there. Anyone else? Well, I've mentioned this a few times already, but insulin and glucagon those are both reacting to glucose levels in our blood, right? And if hormones are traveling through our blood, then right now in the human trials, really, we measure the hormones in blood. And when we get to marine mammals, we often will also look at blood. All right, now this works for captive marine mammals, and it can also work for seals and sea lions, wild ones, if you're able to actually capture them and kind of put them under and gather samples that way. It doesn't work so well when you have these wild whales and dolphins that you're trying to get data from, because we can't ask them for blood. So here, this is at SeaWorld. Oftentimes with any of the captive marine mammals that they will take blood and just kind of check and see how these animals are doing. They will check things like their insulin levels, their cortisol levels, aka their stress levels. They can also see if these whales are pregnant. So they can check their progesterone levels. If those are really high, then that might indicate that these animals are pregnant. And in fact, what these people are doing right now is taking a sonogram of this killer whale because she is pregnant and they're just keeping track of how that's going. We can also look at hormones in urine and feces. So urine, very difficult to get a hold of in the wild. Feces, you can. A paper just recently came out where they collected feces from a couple hundred, I think, individual right whales, which was really cool. And then they looked at the glucocorticoid in that feces. So glucocorticoids are stress hormones like cortisol. So we had to figure out a different way with these animals to be able to get hormones. And the way that is most common right now is by getting blubber samples. So marine mammals have this specialized fat that's right beneath their subcutaneous fat, and it's called blubber. And that blubber, as you can imagine, is very, very fatty, and that actually is a place that these hormones love to hang out. So a lot of these hormones that we're looking at and that I'll be talking about today are lipophilic. Right, so they love fat, and that's where we're going to find them. So if we can get access to this blubber, then we can look at these hormones. So the way that we can do this in the wild is with a crossbow. So there are people out there, their jobs are to get blubber samples from wild animals, and they have to go target practice with crossbows because otherwise you'll just miss the animal. So you have a crossbow, and it's fitted with this specialized bolt that actually has a stopper at the end of it that acts in 2 ways. 1, so that the arrow doesn't go too deep into the animal, but 2, to also let that arrow float. So you can retrieve it afterwards. And it's fitted here with this steel that has backward-facing tines in it. So what ends up happening is you shoot this, in this case, a dolphin, and that steel goes into the animal's body, and then it pops off because of that stopper, and then it just floats, and then the researchers go and gather it. And then what ends up being in there is skin, which is this dark part here, and then this fat. And then researchers can use that because you don't really need a lot of fat to look at the hormones, but we can look at the hormones from that particular animal. Again, since we know basic ranges for what hormones should look like in a lot of marine mammals, we can get an idea for perhaps how stressed out this animal is, if they're pregnant, if they're not, if they're sexually mature also. But I'm using something different, which is the earwax, okay? So let me talk a little bit about the importance of studying marine mammals. Because especially at Baylor, I often have undergrads that are asking me about, what do you do? Especially the students that I'm teaching. And I'll tell them that I study marine mammals, and they kind of go, why? Why wouldn't you study humans? Well, humans are very hard to study sometimes. But in particular, I've always found marine mammals really cool. So if we think evolutionarily, they were terrestrial mammals just like us, but then they went back to the ocean. And in order to do that, they had to come up with a whole different suite of adaptations to succeed in the oceans. And I think that is the coolest thing. So on top of that, many species of whales are endangered. And this gives us a special double whammy because in 1972, the US passed the Marine Mammal Protection Act, which means we think marine mammals are very special and we're going to protect them more than other animals. You guys know what show was really popular in the 70s? You ever seen Flipper? No? Okay, so the show Flipper, do you know the show Flipper? Oh my gosh, shaking heads. Okay, next time I will include a clip. I will make a note of that. But Flipper was this incredibly popular show in the 70s that followed a bottlenose dolphin that helped kids and families and all of that. It was basically like Lassie, but with a dolphin. And what ended up happening is tons of children actually sent letters to Congress and demanded that marine mammals be protected. And Congress listened, which was amazing. They must have gotten a lot of letters. So that's why the Marine Mammal Protection Act was passed in 1972. And then the next year, the Endangered Species Act was passed. And so what this means is that not only does the US say that we think marine mammals like whales are extra special and we have to protect them, but if they're endangered, we also have to protect them. And so what this means is we need to manage these populations so that they are staying healthy for the rest of their lives, for the rest of existence, as long as the US is around. So we need to look at the size of the population. So if we know the size of the population, then we know if it decreases or increases, if that's good or bad. And then we need to know the growth rate of the population. So if we know the growth rate and it's increasing, that's great. If it's decreasing, obviously that's not good. But it can also let us know kind of the maximum possible rate that a population could grow. And that lets us know, well, I'll get to that later. So the way that we currently get at this information, size of a population, as well as a growth rate of the population, is we follow around individual whales. So there's two kinds of whales I'm going to talk about specifically here. First is the humpback whale, which these are pictures of the underside of its tail, and orcas, which are toothed whales. They have teeth, but humpback whales don't. They have baleen instead. So with humpback whales, the underside of their tails is as unique as our fingerprint is. So this coloration is static, the shape of their tail is static, and even those scars will stay around until they die, until we stop seeing them. So that means that when we see this individual, if we're able to get a picture of the underside of its tail every time we see it, we're able to track it over the entirety of its life, which means that we can say, hey, we know roughly how old this whale is. We know, on top of how old it is, we know if it is a mom. All right, we'll know if it is female because mother and calf pairs stay very close together. And we can track where we see it. So if we see it in one bay 1 year, but then in another bay another year. And the really interesting thing about humpback whales and a lot of the baleen whales is that they migrate. So they go from their feeding grounds to their breeding grounds. So these particular population of humpback whales, they will feed and shove food in their face all day long in Alaska. All right, so in the summer in Alaska, that's when they're feeding. And they stay there for about 6 months, and then they go to their breeding grounds where they're not eating at all for 6 months of the year. And this is when they're mating and giving birth. So we can follow that pattern there. And the orcas, there's some resident orcas, and they'll actually stay, as the name implies, in one area. And they have very special patterns on the side of their bodies. So we're also able to follow those individuals around. But then there's issues like this, where this is what actual sighting data looks like for one particular humpback whale. And you're going to see that there's huge gaps in the data here. All right, so this animal was first sighted in 1975. That's the first time it was ever seen. They didn't make note of what kind of the reproductive state of the animal at the time. And all we really know is that we saw it and where we saw it. And then in 1991, we saw it with a calf. So we learned, okay, it's a female and it had a calf at this time. And again, the location. And then we didn't see it again for 6 years. Right? And then we didn't see it again for 5 years, and then 3 years, and then we didn't see it again until it washed up on shore dead. So this is the kind of data that we're able to get with long-term observational data sets. And it's not the best data. But until recently, it's the only kind of data that we've really had. Until the earwax. So we wanted to know if these earplugs, this buildup of earwax in the ear canal of the whale, we wanted to know if it could provide us some of the data that we've only been getting up to this point with observational data sets. So these have traditionally been used to age an individual whale. So you see these layers here? Do you know of any other kind of biological material that has layers like this? Yeah, exactly, tree growth rings, right? And it works in the same way. So remember I was telling you that these whales, particularly baleen whales like humpback whales, that they're migrating, right, from their feeding grounds to their breeding grounds? Well, it turns out that these light layers that you see are put down when they're in their feeding grounds, because the cells that are in each of these layers during the time that they're feeding are actually full of fat, which actually makes those cells appear lighter. All right, and then these dark layers are put down while these whales have traveled to their breeding grounds and they're not eating anymore. They're fasting during that time, so those cells are not nearly as full of fat anymore. And that's why they appear darker. And so since we know that timeline, one dark layer and one light layer means 1 year of time, we're able to age from these earplugs, which is really cool. So that's a great first start for how we're using these earplugs, but we wanted more. So in 2013, my advisor, Dr. Stephen Trumbull, and his colleague, Dr. Sasha Yusenko, figured out that they can actually get hormones and even contaminants from each one of these individual layers that relate to how that animal's life was at that time. And that's really incredible. We've never been able to do that before. And when they published that paper, they published from one animal, and that's it. And they got it in a really good journal too. So that just to show you how incredible that discovery was. And we know that each one of these layers is 6 months of time. So if this animal here was 40 years old, we actually have 80 data points for it, where we can look at hormones in particular. And the wax that's up here is the wax that the whale put down when it was a calf. And the wax that's down here is the wax that the whale put down right before it died. So we can actually assign an age and even a year to each and every one of these layers, which lets us put this, puts this data into time, into human time, but also in age for these individuals. So really what I wanted to get at here was to study, I want to study these population dynamics so we can provide better data. Because the data I was showing you before with those observational data sets where there's huge gaps in the data, that's not great data. We have better data now. And with that data, I want to input those numbers of population dynamics into models so that we can get a better idea of how these populations are doing and how they might do in the future. And even we can use these to calculate allowable removals. So this is a government speak kind of thing, but allowable removals means how many animals do we think this population could lose a year and still do well. So if we think that number is 100 right now, with my data, we might actually show, you know what, 100 is too high. We actually need to make it lower, like 50. And this is not so much like the US government is going out and killing whales, but how many whales are dying? Sometimes there's shipping accidents, right? There's issues with choking on plastic bags with some animals. So I'm really interested in kind of inputting into that. So I've mentioned several times already baleen whales, and I wanted to just talk a little bit about what those are. So everything with the star is a baleen whale. So that would include the blue whale, which is the largest animal to ever roam the planet Earth. And then the fin whale, which we call the greyhound of the sea. It's extremely fast, and it's the second largest baleen whale. And then we go all the way through until the smallest baleen whale, which is called the minke whale. And they're called baleen whales because they have these plates of baleen hanging from the top of their jaw. So in utero, they actually do have teeth, like us, those teeth fall out and then they grow baleen instead. And the reason they have baleen is so that they can feed. So we have these largest animals in the ocean, and instead of them feeding on the next largest thing, they're actually feeding on the very smallest thing in the ocean, which is zooplankton and small schooling fish. And what they're doing is they can take a big mouthful of water that has that prey item in it and then strain it out. So it's like when you put spaghetti into a colander to separate the spaghetti from the water. It's the same idea. And so then instead of swallowing all that salt water with their food, they just get the food. So these earplugs are located really far inside the head of the whale. So they're growing in the external ear canal. So our external ear canal, right, is open to the environment, but theirs isn't. They really don't want water in their ears. And so the weight of their blubber and their muscle actually collapses the outer part of their ear canal. And so what that means, so if we take this whale and flip it upside down, that's what we would be looking at here. And see these pink structures? Those are the ears of the whale. So, let's see, I can point. So this here, we call that's the ear bone of the whale. We call that a bulla. And if we take a closer look at that, you see that finger-like projection coming off of that bulla? That's the earplug, all right? Now, the brain of the animal is just on the other side of this. So we can't actually extract these earplugs without killing the whales, all right? Which is why we don't kill the whales anymore. All right, the Marine Mammal Protection Act says we cannot kill whales, all right? So what we have to do is get earplugs from museums. So it turns out because these earplugs look so weird and so interesting with all those layers, that museums have actually kept a hold of these for decades. So our oldest earplug comes back from when it was first harvested in 1910. And when I aged that whale, we actually found out it was born in 1856. So that means we have over 150 years of hormone data from these animals, which is incredible. And then as for newer earplugs, we actually partner with several stranding networks across the US. So when they get large baleen whales who have washed up and they're dead already, they will actually extract the earplugs for us. But it's a very long process, as you can imagine. You have to get through several layers of bone, maybe a feet or two of tissue actually, because that blubber can be very, very thick. So it can take 4 hours if you know what you're doing to get us an earplug. So they're very, very valuable tissues. So here is one that I've aged, and that is a little difficult to see, but I've actually counted up each one of these layers with these little red lines, and so this individual is 44 years old. And if you can see this, it's only 5 centimeters long. All right, so it was that big. 44 years of data in that small of a space. All right, and if that's 44 years of data, that means that it's 88 layers that I have to go through and cut. All right. So the kind of metadata that we get with these earplugs, we get year of death, we get the location where they died, their sex in most cases, species most of the time. There are a couple of plugs we have where we don't have species, and age. And age is when I age it. So that's something we generate when we receive the plug. Sometimes we'll find that whalers at the time tried to age them, but it's really, really wrong. So what I want to do, if I want to get hormones from each and every one of these layers, I have to cut each and every one of those layers apart. So I do it with a razor blade and reading glasses, and occasionally a magnifying glass as well, because they are so small. I will cut off these little bits of wax. We'll homogenize them so that wax is now all the same consistency. And I will go ahead and extract the lipids or the fats from that wax. And the reason I want to do that is because the hormones that we're interested in, again, they're lipophilic, so we're going to find them in the fat. And then I will do an ELISA. Now this is an enzyme-linked immunosorbent assay. It's a pretty common assay in hormone like endocrinology labs. And what it's doing is it's telling me what the hormone concentration is of each and every sample that I put into that plate. And each one takes about 4 hours to run. And I'm skilled enough now that I can do 4 at one time, but it makes my arm hurt because I have to pipette a bunch. All right, so let's get into the data here a little bit. So since I'm interested in population dynamics, I'm actually only interested in females. That's just how it goes. So I got hormones from 11 females, and they ranged in age from 13 to 58 years old, which is really incredible. And these whales lived from 1902 all the way through 2014. So that's over 100 years of data. And overall, I looked at more than 700 individual layers of data, which took a while. Now, we like to show this timeline to kind of show you how old these whales were and from how far back we're going. So you'll see that we looked at 4 different species: fin whales, humpback whales, blue whales, and minke whales. Now we have 5 fin whales, which is really great. 2 of them are quite young. And these 3 humpback whales all lived a fairly long amount of time, which is really awesome. And they're all from recently stranded individuals, which is why they're pretty far forward in the timeline. And then those last 3, the blue whales and the minke whale, also died in the 60s. So all of these species except the humpback whale we got from museums, which is really awesome. So the Smithsonian Institute, as well as the London Museum of Natural History. So the hormone in particular that I wanted to look at is progesterone, because I wanted to see if we could determine how many pregnancies an individual whale is having over her lifetime. Because this is something we haven't been able to say before because of all those data gaps that we see with this observational data sets. So first off, progesterone is lipophilic. Great, we're going to find it in fatty tissue. But also it is a key indicator of pregnancy in quite a few animals. Most animals, in fact, that I was able to see, most chordates. And this is what it might look like if an animal got pregnant. So this is a graph for a dolphin. We have weeks post-conception on the x-axis, and then on the y-axis we have progesterone concentration. And so post-conception means, at 0 is the time when this whale got pregnant, or when this dolphin got pregnant. And 5 is 5 weeks later, where we see this huge increase in progesterone. All right, and those progesterone levels stay fairly elevated until birth. So we were hoping to see something like this with the earplug. But instead we see something like this. All right, and the reason that it looks so messy and there's some weird peaks and sometimes there's flat areas is because each one of these points is 6 months of time. And the gestation time, or how long a whale is pregnant, is only 10 to 12 months. So instead we thought, why don't we look at this data in a different way? Instead of looking, we'll still look at it by age, but instead of looking at it by progesterone concentration, why don't we look at it in a way that will help us see where we see this big change in progesterone concentration instead. So that's in fact what we did. We looked at change in the progesterone concentration from one layer to the next. So this is the same graph I just showed you, and this is after we normalize it in that way by looking at % change in progesterone from the point before. And it looks a lot clearer now. We see these big pulses in changes in progesterone, and then we see these other areas where the change in progesterone is not so intense. And we think these are what are correlating with actual pregnancies. So I thought, well, we actually have 3 humpback whales that we know were alive recently, and they're from a population that people follow around and get observational data from. I wonder if we have any sighting data for these animals. And we did. We had sighting data for 2 of them. And actually, I already showed you sighting data for this specific animal earlier on. So each one of these arrows indicates a time when this whale was sighted. And these first 4, it was sighted with a calf. So sighted with a calf, pregnant. Sighted with a calf, pregnant. Sighted with a calf, pregnant. Sighted with a calf, not sighted with a calf, not pregnant. So they match up really, really well, which gave us a lot of confidence in knowing that when we see these peaks in progesterone like this, peaks in the change of progesterone, that those do indicate pregnancy. And that was a huge thing for us because now it means that we can actually calculate population parameters from this, population dynamics. So this let us calculate pregnancy interval. And what researchers are publishing now is they're publishing calving interval, meaning how often do they see a whale have a calf and how much time is it in between that itself. So here for fin whales, we see that the calving interval is usually a whale will have a calf every 2 to 2.7 years. And our data, that matches our data really well. We see that fin whales are having a calf every 2.6 years. And you can see that that matches really well for these first 3 animals, these first 3 species rather, and not well for the minke whale. Now, the reason it doesn't match for the minke whale is because the minke whales are not well studied. Because they're the smallest of the baleen whale, I mean, they're still huge compared to us, but they're the smallest of the baleen whale. It makes them very hard to see when you're out in the wild looking for whales. So the research here, this publication that I found that says that calving interval is every 1 to 2 years, that publication is actually from whaling data. And whalers at the time, they couldn't actually kill whales that were currently with a calf, which ends up biasing the data hugely. And it means that this calving interval is really twice as high as it should be when we're looking at our pregnancy interval here. Now, if we look at pregnancy rates and compare that to birth rates, we see a similar pattern here, where we see that the birth rate literature matches pretty well with the pregnancy rate. But again, minke whale, that doesn't add up at all. But again, that's because this one publication I was able to find for the minke whale, again from whaling data, so it's really biasing that estimation up. So our data, I would trust our data far more when it comes to that. And again, all these birth rate, all this literature for birth rate and for birthing interval, for calving interval, they're all based on that data that has huge gaps in it, right? With these earplug, from the data from the earplug, we don't have these kinds of gaps. All right, so for population dynamics and really to actually calculate and look at a potential growth rate for the population, I wanted to calculate the maximum rate of increase for a population. And that means barring any kind of outside influence to a population, what can this population possibly grow at the absolute max if they had everything going for them? So there's some factors that I need in order to calculate rate of increase. And it turns out I'm able to actually look at this with pregnancy rate and age at first parturition, which means the age at which they first give birth. All right, so I can get that from the earplug, which is really great. So this data is all generated from the earplug for these species. And then I also need survival rate, and I can get this from the literature. Not nearly as reliable, but I can't produce survival rate from the earplugs. It's just not what we can use them for. So when we do this calculation, and I'm not going to show you the equation because it's complicated and long, so you're welcome. But I need all these parameters, sex ratio at birth, which we just assumed to be 50-50. All right? And when I compare the numbers that we got to another publication, which got a mean of 8.6%, our numbers for humpback whales are right in there. So that made me really happy. And so I went ahead and calculated that for the other 3 species. So you see for fin whales, it's 4.2 to 4.9%, blue whales 7.5 to 10.1%, and then minke whales is a negative number. But again, that's because minke whales are not well studied, and the survival rate for minke whale was quite low. All right, this is amazing. We've never been able to get rate of increase from baleen whales really before this point with such good data. It just hasn't been done before, and that's really the true value of these earplugs, is that they let us ask so many questions that we've never been able to ask before. But the problem with the model that I just showed you is that it's assuming that these animals are having babies basically their entire life at the same rate. And I wasn't sure that was particularly true. So in humans, right, females don't have babies the entirety of our lives, right? Menopause hits around 50 or 60 years old. And so I thought, surely with these baleen whales, they must also experience reproductive senescence or menopause. But the literature wasn't showing anything. And it's because we haven't been able to even ask this question before. And so I asked it. So here we have age in year on the x-axis, and on the y-axis we have percent change in progesterone from baseline for each of these individuals. And we see this very telling pattern here, where we see this increase from ages about 5 until 20, and then a kind of a plateau from 20 to 40, and then it falls again, 40 and on. And this is classic signal of reproductive senescence. All right, so this is for fin whales. I was blown away. But for humpback whales, we don't see that. We don't see that at all. And it could be that we don't have whales that are old enough yet, and maybe we're not seeing it, or maybe there's something special about humpback whales that lets them not actually experience reproductive senescence for whatever reason. A very interesting question. And actually, humpback whales worldwide are doing quite well, and they're recently delisted from the Endangered Species Act, which means that they're not endangered anymore, most populations. So I wanted to incorporate reproductive senescence now into my model. And I wanted to do that in a way that I could also project into the future. So I wanted to get a rate of increase and then compare that to what the other model was saying for the rate of increase. And so I'll just go ahead and show you what that looks like. So we have year on the x-axis where I projected forward until 2050. And then we have fin whale population in the thousands here. And so again, this is just assuming, just if we left this population alone, how would it do? So if we don't take into consideration reproductive senescence in fin whales, we get up to about 220,000 individuals at 2050. But if we do include reproductive senescence, we only get up to about 155,000. And that's actually a 50% difference, which is really huge if people are actually using these numbers, the non-reproductive senescence numbers, in their population models. A 50% reduction could mean huge differences here for incorporation into population models and for those allowable catches I was talking about earlier. So really, we need to use reproductive senescence in the model when we're talking about fin whales, but maybe we don't when we're talking about humpback whales. And that's really interesting, and maybe we should manage them in different ways because of that. So just very quickly, I think we have a few minutes. I'm curious, I only talked about one aspect of what we're doing with these earplugs. Can you think of other questions you might ask with these earplugs, knowing that we can get data, hormone data, contaminant data from these animals over their entire lifetime?
Maybe how they died.
How they died?
Disease or stress related or...
Yeah, that's a really great question. Yeah, so if we looked at the most recent layers, we could see if they might have had higher stress levels or if maybe their contaminant levels went through the roof for some reason. Yeah, and then we might be able to make some hypotheses about why they died. Any other parameters, any other, maybe any other kind of particles we might be able to see in the earplugs? Yeah, certainly, yeah. So the contaminants we are able to look at are like pesticides, absolutely, DDTs left over from like old fridges. There's a ton of contaminants. Actually, one of the people I work with, Zach Winfield, he's working in a chemistry lab, and so he helps us with that aspect of the research. We even look at heavy metals. We can also look at trace elements. This is really cool. It turns out that a mammalian body actually needs a lot of zinc during pregnancy. And so we can actually look at the trace elements in the earplug layers. And if we see a decrease in zinc at the same time that we see an increase in progesterone, that helps us confirm even more that we are seeing a true pregnancy. Are there any? Oh, go ahead. Yeah, that's a really great question. So the question is, can we determine the environment that the animal lived in with just the earplugs? And we absolutely can, and that's a stable isotope question. So with stable isotopes, we can actually take very, very small mass numbers from each one of these layers and determine what the stable isotopes are. So we can look at carbon and nitrogen stable isotopes, and that lets us know if we see these similar patterns for an animal that we know was living in the Atlantic Ocean. Then we have this other animal where we don't know where it lived. If those match, we know that this other animal also lived in the Atlantic. And we could possibly get more specific than that, but right now most of our earplugs are we only know ocean basin wide where they lived. Great idea.
Could you use it for further cloning if the population had been destroyed before and you only have the wax structures, maybe have a little bit of genetic material on it? Could you use that for cloning?
Yes, that is a fantastic question. And actually, my lab mate there, Brooke, who came with me today, she is going to be looking at the genetics of these earplugs. In particular, she's really interested in the epigenetics of the plugs. So epigenetics is basically, as we're living, right, we all have, so in my body, in each one of my cells, I have the same DNA, all right? But as I'm aging, there might be certain environmental factors or certain hormones, like cortisol, that will actually change which genes are expressed. And so what Brooke is hoping to do is look at the entirety of that earplug from when it was a calf, when it was a juvenile, early adult, late adult, and then right before it died, and see how the DNA expressed might have changed, which could definitely tell us some super interesting things about their life. So yeah, we're very excited to have her in the lab. All right, well, I just want to open it up to general questions now. Any questions about anything I do, how I got here? I drove. Yeah.
Does being shot with a crossbow hurt a dolphin less than it would hurt me?
Ooh, so that's a great question. So the question was, is a dolphin going to be hurt more or less than if I shot him with a crossbow bolt? So this is the thing. Their blubber layer is really thick. So that stainless steel plug that I was showing you guys, that is only going to go into their blubber and it's not going to go into the muscle. If I shot you with that, it would definitely go into the muscle and it would hurt a lot more. And there's been a lot of studies about how much biopsy sampling dolphins and whales, how much it bothers these animals, and it really doesn't seem to bother them much. And when we're thinking in particular maybe about infections, very, very few animals have ever died or gotten an infection from being biopsy samples. So it's a pretty great technique to use if you're trying to get samples from wild animals and keep them alive. Okay, yeah, that's a great question. So the question was, is it expensive to get the earplugs from museums? And it turns out they are begging to give them to us. All right, and the reason is because museums, until very recently, have been starting to fall out of fashion. They were just these kind of reliquaries of these old samples of tissue, and like you could just go and look at weird stuff and cool stuff. But museums really want to be part of science again. Not that they ever really stopped, but they're making more of an effort now to reach out. And we're actually, scientists as well, are making efforts to reach out to these museums as well. So they actually donated them to us for free. And so they're hoping, and we're hoping, that we can just keep that relationship going. So they look good because they're giving us these really valuable tissue samples, and then we look good because we're able to use those. So we work together really quite well. Yeah. All right. Well, I think, yeah, it's 11:15. That's all I've got. Thank you all.